Hello, everyone. My name is Hillary, and this is part 3 of clinical research terms from the ICH E6 R2 glossary. And we will be going through some terms. If we come across anything that has documents that we want to look into, we can use the DIA reference model and kind of look up where those documents would live to give you an idea in terms of the relevant of the items in the glossary to actual TMF clinical research documents. Okay, so we will jump in and just go straight to the actual guidelines that we've been working through. And here we go. So we were left off. So we've been going through the glossary, which kind of starts on this page 8. And we've looked at, last time we looked at informed consent and talking about investigator brochure. And I believe we left off talking about the protocol. And then I'm just going to kind of skim through here, slowly scan through items so that you can kind of see these. I recommend that you take the time to go through this link that I sent out and actually look at these items and start to get yourself familiar with the different terms. And then also go through the DIA reference model, that link that we have, that also has a document library attached. So you can start to get familiar with the documents that relate to different artifacts that kind of, and then come back to here and you can relate them to the actual clinical research term. Okay, so we have QA and QC. And so just kind of want to look through those. That are common terms within clinical research. So QA is quality assurance. And then QC, and I'm actually curious to see what the ICH, which is kind of like a regulatory or a committee board that's international, and kind of what their terms are and the difference between what they see of quality assurance and quality control. A lot of times what we talk about with documents is quality control, but let's go ahead and read their terms. So all those planned and systematic actions that are established to ensure that the trial is performed and the data is generated, documented, and reported in compliance with GCP and applicable regulatory requirements. So that's their definition for QA, quality assurance, quality control. The operational techniques and activities undertaken with the quality assurance system to verify the requirements for quality of the trial-related activities. So it's interesting that they're talking about more of QCs, like the techniques and the activities undertaken to assure quality assurance. So quality control. We think about quality control in terms of documents. You're going to QC the document. You can do a quality check. And so those are some different terms there. Okay, randomization. This is also important. We talk about patients being in clinical trials, and it's important for patients to have their rights protected and that things are done in a controlled manner and unbiased, right? So if you had a study of 100 patients and you did a certain location, then you might have a bias in terms of like if that demographic has a certain financial status or that demographic has a certain ethnicity, then it wouldn't really be fair. It wouldn't be able to use that data to say, okay, this drug works great, but maybe only just in that small population. So that's why usually they like to have like global and even like if you're doing the United States, within the United States, very a lot of different sites and different locations. And this is kind of just a background on how the studies are run. And the idea is that you're going to randomly, if you have a controlled study, so that means that you're giving one group the actual investigational product, right, which is IP, which we talked about previously. And then you're giving another group as your controlled group. So sometimes ideally they don't get anything, but usually they'll get a very similar version of the drug, but you call it placebo. So it's either like saline or a sugar pill or something. So that way people think they're in the study, but they're not actually, because it's been proven that the act of thinking you're getting treatment actually does help, right? So if you take a control of just normal people who don't think they're getting a treatment, they don't think they're improving, that's actually, you'll see a higher result of people taking the treatment doing better. And you don't want that to be incorrect in the sense where it's because they're excited and they're trying to get better. And so randomization is it's assigning the patients to either the controlled or, let's say, different arms. That's another term, different kind of groups within the clinical study. And so the idea is that it's not somebody just kind of doing it. It's very random, right? It's not, no one's being able to bias it and be like, oh, I want the study to be more successful, right? Because I'm the company and I have millions, maybe small billions involved in this, and I want this to be successful. So therefore, we're going to make all the people who are less sick in the treatment group and all the people who are more sick in the control group, and that way the treatment will for sure do better than the control, right? Which is not good, right? That's a bias and an unethical study, right? And so randomization helps eliminate that, right? So their definition is a process of assigning trial subjects to the treatment or control groups using an element of chance to determine the assignments in order to reduce bias. All right, regulatory authorities. We've talked about this. Bodies that have power to regulate. That's a general term, but right in the ICH GCP guidelines, the expression regulatory authority includes the authorities that review submitted clinical data and those that conduct inspections. These bodies are sometimes referred as competent authorities. Okay, now our first thing that we went into on part 1 of this week's series was we talked about SUSARs and MedWatch forms and kind of the idea behind an SAE. We were just looking at an AE because we started at the beginning of the alphabet. But here we'll just quickly touch on an SAE is serious, and it's any untoward medical occurrence that at any dose, right? So you have to see that the dose doesn't matter, but it's the result. It's resulting in death, life-threatening, requires inpatient hospitalization, result in persistent or significant disability, right? So like maybe they don't have to be hospitalized, but now they can no longer walk on their right leg. That's a significant impact to their daily life, right? So that is a significant adverse event. They didn't lose their life, they didn't get hospitalized, but you've kind of, especially if it's permanent, you've potentially disabled this person or caused a disability in their life, and it will hinder them, right? Is a birth defect. And so we can go into more. I think it'd be great for us to dive into the rest of the document, exploring beyond the glossary to really learn and study this document. But that's really important. And if you were getting ready for a CTA job, you should know this. That could be a good interview question. What defines or differentiates between an AE and an SAE is death, life-threatening, hospitalization, disability, or birth defect. So good. When you're doing these things, especially if you're getting ready for a job, you should always be thinking, how can somebody use this information to test me? Like how could I prepare myself for an interview, and also just how do I use this in a day-to-day job within clinical research? Okay, source data. All the information in original records and certified copies of original records of clinical finding, observation, other activities in clinical trial necessary for the reconstruction and evaluation of the trial. So we talked about how the TMF, the idea, and as it's mentioned here, is through the trial master file, somebody who's what we talked about, regulatory authority, can recreate and really understand what was occurring within the study. So, and that's why it's important to have these correct documents all organized so they're easily found, right? That's why we want to put items into the correct artifact, have a naming convention, note which documents go into what, so that it's all consistent. Consistency is really important. Okay, now we've talked about source documents. It's kind of basically very similar to the source data, talking about documents, data, records, and we also can include photos and things like that. We talked about sponsor, right? Sponsor is the the person who's running, or not the person, the company that is running the trial. They are sponsoring the trial. Now, that doesn't mean they have to do everything. They are allowed to delegate out responsibilities, but it's really important that they always maintain responsibility and management, right? So you could have a CRO and you could say, oh, the CRO is managing the CRAs, right? But in general, the sponsor is responsible for making sure that it actually occurs properly, right? Like the CRO is being hired to do it, but in the end of the day, the CRO is like responsible to get the job done, but not responsible in the sense of like, did it occur correctly? Is it ready for filing? Now, the CRO should do a good job, but it's the sponsor's responsibility to manage the CRO, and they have to provide proof of that. And that's why you want to file your meeting minutes with the CRO, so that you can show the vendor management and show that it was done correctly, right? Okay, so the sponsor investigator, this is a little bit not super common, but an individual who both initiates and conducts alone or with others a clinical trial, whose immediate direction direction the investigational product is administered to. Okay, so this is a kind of a more of a one-off situation in terms of what we're talking about, where we're like looking at trying to help people get jobs at sponsor companies. So this is a different situation where the sponsor and the investigator are like the same. I'm trying to think of a situation I think this could be applied to is if, for example, like UCSF does their own studies. So they're both the sponsor and potentially the investigator, right? So like they might have an investigator, but the investigator is from UCSF, and UCSF is kind of like the sponsor, and the investigator's kind of in charge of running everything. Whereas usually you'd have something like a big biotech that then hires out UCSF as the investigator, like, and the investigator is the person who's running the trial, the head PI, the head principal investigator, and then the institution, UCSF, would be the institution, right? And or like the site. Okay, we've talked about SOPs, standard operating procedures. Maybe it would be good to look at some kind of like, that could be a good future meeting to look at some very basic standard SOP template on like the TMF, so you guys can get understanding of what an SOP really is. But it is a detailed written instructions to achieve uniformity of the performance of a specific function. As it mentioned, it's really good, for example, when you have if you have like you're filing a TMF document, and how you file it, and like where you put it, and what the process is, who do you send it to, in terms of like, are there 2 steps in terms of the QC? That would be documented so that like ideally there is uniformity between the different studies. So like one company has multiple studies, and the people that are processing documents and people who are running the study too, but for the TMF sake, there are going to be TMF SOPs that describe what you're doing, and kind of what is the process at this company. So the SOPs are company specific. Okay, sub-investigator. We talked a little bit about this, but so these terms should be familiar to you, but this is basically the non-head doctor, right? So you have a PI, a principal investigator, and then you have a sub-investigator. Usually you might have multiple sub-investigators, and certain places will cap, like they'll say, okay, you can have one principal investigator and 2 or 3 sub-investigators. And so there's a whole bunch of different reasons behind that, but usually if you get too many, which can actually, it's a common trend in the US, where they'll have lots of investigators, and that can make it so that people aren't as familiar with the study. It works if they're seeing tons of patients, and yes, you need lots of people to help support it. But if they're not seeing tons of patients and it's not a lot of work, then there's no reason to have 10 sub-Is, because that means that each sub-I gets less interaction with the patient and like less familiarity with the study. Okay, so sub-investigator is kind of the level, like is the sub to the investigator. So you have a principal, PI, and a sub-I. So there are terms to know. Subject and trial subject. individual who participates in the clinical trial, either as a recipient of the investigational product or as control, which is what we talked about, right? Subject identification code, a little code given so that, because you don't want to have the subject name, so that everything is blinded, so that the people don't know exactly who's getting what, right? And I'm going to kind of jump around. I think we're almost done. Okay. But the trial site we've talked about, an institution is the same thing as like a site. And then that's where the PI and the sub-I are, whereas the sponsor is the company with the medication typically. Okay, unexpected adverse drug reaction. So this is what we talked about, where there is an adverse event, right? Something that's not good, and unexpected is worse because it's not in the IB. So here we go. So it's not something that they thought was going to happen, right? So I talked about a long time ago working on a study where they didn't know, and maybe they had seen some traces of this, and like, because you're testing it in the lab first, and then you test it in humans. And this is the first time, depending on what phase you're in, if you're phase 1, it could be the first time that humans are getting this medicine. And usually it doesn't get to that stage unless people think that it's going to be really helpful and be safe. But sometimes things happen. And for example, the one that I worked on, there was something called alopecia, where you like lose your hair. And it was just it just happened. And it was very subtle and not everyone knew it. And then once they found out, they did need to update the IB. Right. And they also needed to update the ICF, which we talked about last time too. But here it's saying unexpected means that it was unknown, basically not consistent with what they thought, what was already in the IB. And then we just talked about SAE, as in severe, right? And so a SUSAR, which is a common term, is a severe unexpected adverse drug reaction. So it's a good term to know, like what does SUSAR mean, right? And so then you can respond, well, it's severe, which we talked about, like death, hospitalization, disability, and birth defect. And then it's unexpected, so it's not in the IB, and then it's an adverse drug reaction. All right, vulnerable subjects are individuals whose willingness to volunteer in a clinical trial may be unduly influenced by expectation. And so they might think they need to be in this trial, or they might be, so you definitely want to watch out for this. Examples of members of the group. Okay, so, and then basically just all other vulnerable subjects include patients with incurable diseases, people in nursing homes, unemployed or impoverished patients. So they kind of they might be joining for other motives, kind of a thing, and or like it might not be ethical to have them, like homeless people. Like, do they really know what's going on, or are they just seeking medical treatment? And so it's basically anyone who's also incapable of giving consent. That's not okay. Or usually you have to make sure, like, let's say there's a parent guardian or somebody who's really in charge of speaking for them. But all of those you want to be very careful with, right? Because within clinical research, you want to follow ethics because the overall goal is to help people, is to provide and create medicines that people need, right? And so you want to make sure that we are protecting people in the process. Okay, well-being of of the trial subjects, physical and mental integrity of the subjects participating in a clinical trial. So their well-being is important. Okay, and this is actually kind of interesting. We are going to go through, just these guys, the addendum. Sorry, so we talked about how in the very beginning that this is the R2, version 2, right? It's been 30 years since the first version, and this document, these 66 pages, is really important to know and understand, because this is basically what the world believes is important for clinical trials. And they've tried to condense it really tightly, and just here's the top things you need to know, basically, to run a clinical trial. And over 30 years, these are the 3 things they've added. So they've really tried to keep it lean. And the 3 different new glossary items. So this could be an interview question. I think it'll be a little intense, but you know, you gotta be ready for everything. And it could be, what are the 3 items, 3 new terms added in the ICH R2 or R6. And we'll go back to this so you can see the terms. I want to say E6 R2, and then I want to. This is kind of the what ICH means, and then this is what I was looking for, the technical requirements, and then to basically run human studies, right? Okay, so these are the 3 items. And certified copy, we talked about this. This is a copy of basically making sure that this is, while it's not the original source, we talked about this, not the original record, it's now basically acting in a place, and the person can like certify it. The person who's uploading it say, I do certify this. This is an exact copy of the item. And so some places, when they have you upload documents into the TMF, they'll have you do a little drop down and say, I certify this copy, right? Because I'm scanning it. What's in there is digital, and what I have here is not. And so I have the original, but I certify that this copy that's online is correct. And ideally at that point, the person should triple check that all pages got scanned, everything is good, everything's captured, because sometimes you might destroy that original, especially as we move into more of a digital age. So yes, that's a certified copy. So it's interesting to see that of the 3 items, basically 2 of them have to do with like systems and TMF. The middle one, monitoring plan, we talked about monitoring when a person, a CRA, goes out to the site. But now it's very common to have a monitoring plan. So that it's basically describing the strategies, the methods, the responsibilities, and the requirements for monitoring the trial. And so that would be a plan, like we talked about, where the sponsor has the overall like responsibility of making sure everything runs well. And so they produce a plan, and they ask their vendor, the CRO, to follow the plan. And that's part of their like management process, and proving that we are trying to manage this and trying to make sure that the study is conducted appropriately is by having in place a monitoring plan. So very specifying what exactly they expect, how quickly should reports be made and submitted and reviewed and like finalized, and who's going to review the report. Is it just going to be the CRO? Typically, it should be a sponsor review. Most of the time I've seen it is where the CRA goes out, sees the site, talks to the doctor, makes any notes on documents that need to be collected or things that have gone wrong or ways to improve the study, which is very important for the sponsor to get that insight, right? They write their report. Then within the CRO, usually there's like a manager, sometimes it's like a CTM title. Usually I found it more so to be a PM title, but I feel like most of the industry is shifting. And then you have a that's the correct. So then the PM is reviewing that report, whatever kind of management authority within the CRO reviews that report, and they should help think about what are things that they can fix at the site. They can tell the monitor, hey, did you catch this? The report previous, you said X was an issue, but now in your new report, I don't see anything about it. Did it get resolved? Did you forget? You know? And so that's when the PM can kind of have like a one-to-one kind of trying to resolve issues in a sense before the sponsor is really aware in a way. That way everything's in the report. And so the sponsor should review the report, and they should also say, they talked about X last time, but they didn't talk about X this time. But a lot of times they don't look to that level. And so that's why the PM would want to know and almost resolve it. So when the manager at the sponsor says, they didn't talk about this, and asks the PM, that goes, oh yeah, actually they just left it out, it's been resolved now. So that it's almost like they're trying to be one step ahead of the process. And that's what we should do in general with work, is always be be very proactive and always be aware and informed, and make sure that you have kind of all the answers ready in case somebody does ask you something. You're like, oh yes, I don't need to like look into that, I know the answer. Okay. This is the last term. We've done a pretty good timing for our 3 sessions on the glossary. The last term and the new term is the validation of computerized systems. It makes sense that they added that within the 30 years because that's something that's new. A process in establishing and documenting the specific requirements of a computerized system that can be consistently fulfilled from design until decommission of the system or transition to a new system. The approach of validation should be based on a risk assessment that takes into consideration the intended use of the system and the potential of the system to affect human subject protection and reliability of trial results. So that's our last item, and that's the TMF is a validated system, right? So if you're talking about the eTMF, right, using Viva, which we went over, I think last week, it's validated. And so a lot of times, certain areas are not validated. That desk drawer in somebody's office, that's not validated eTMF or a TMF system. So that's something to think about. Hopefully you've enjoyed all of these terms and given you a better insight into clinical research by reviewing this glossary. I would recommend that you do a GCP certification program. There's lots of different free ones, and I've put a link in the emails that I've sent out. I also have a link to this document. Later, I'm going to scroll up and we can take a quick look at the table of contents. I think it's up here. Yes, okay, so basically I just wanted to do a quick scan so you can see all the different things. So we just did the glossary section, the 2 to whatever 8, and then now it goes into all these different sections about, and it gives you an idea of what the responsibilities are, right? Responsibilities, what they're supposed to do. So you have IRB, the investigator, right? The sponsor, which is typically where I've worked. It's usually for the sponsor. And so this is really critical. You can learn all about different trial design, record access, there's safety, where we talked about adverse, a whole section on monitoring, a whole section on auditing. And then there's protocols and protocol amendments. And then the IB we talked about. And then essential documents. We'll do another training on this. We've talked a little bit about that. And so this is what they feel are the key documents, the minimum that you have to have, right? And we touched on this in one of our kind of early presentations on TMF. But yeah, hopefully you enjoyed that, and that gave you a good idea of different clinical research terms, and hopefully this has piqued your interest for you to dive deeper and see if you want to pursue a career within clinical research. So yeah, thank you. Please let me know if you have any questions, and have a nice day. Thank you.
Thank you.
Bye.
Bye.